So good day everyone. So for this week's lesson, we will be discussing about isolation and identification of Staphylococci, specifically focusing on the Catalase test and Coagulase test. Okay, so this is our learning outcomes. So at the end of this lesson, you should be able to understand the difference between catalase reactions and coagulase reactions that is used to characterize and identify Staphylococcus species. Also to acquire knowledge on how to appropriately perform the catalase test and coagulase test in identifying unknown samples and explain the biochemical basis for the test results. Okay. So to start, what is Staphylococcus? So Staphylococcus is under the family Micrococcaceae. Okay, so they are characterized by a round shape, so coccus or cocci in shape, gram-positive, and found either as single cell, in pairs, or more, frequently in clusters that resemble a bunch of grapes. Okay? So they are widespread in nature, although they are mainly found on the skin glands, and mucous membranes. So they are part of our normal flora. So we have different types of or significantly important na species ng Staphylococcus. Okay? So they have different characteristics. So this is based on pigment production in low fluorescent serum slant. Okay, so si S. aureus, they have golden pigmented colonies. Si S. epidermidis or S. epidermidis or S. albus, porcelain white colonies sila. And then si S. citrus, lemon yellow colonies. Okay, so citrus, yung name, lemon yellow. So madali na siyang tandaan. So, based on pathogenicity naman, si S. aureus lang ang only pathogenic species among the Staphylococcus family. Okay? So, si S. epidermidis, S. saprophyticus, they are both non-pathogenic. Okay? So, saan nakikita si S. aureus? Usually, infections, for example, boils, osteomyelitis, endocarditis, food poisoning, toxic shock syndrome, tsaka writer's disease. Okay? And then, yun. So, sila non-pathogenic sila ika-classify, although they can also cause infection, usually sa mga immunocompromised individuals na. Okay? Or kaya naman, hospital-acquired infections na. For example, sa prosthetic or indwelling devices ng mga patients. Sa saprophyticus ang pinakasikat sa kanya, it is most common cause ng UTI in young females who are sexually active. Okay? So si S. saprophyticus ang most common cause ng UTI in young females who are sexually active. Yan. So based on coagulase production naman nila, okay, so we can differentiate them. Ang only coagulase positive lang is si S. aureus. So, S. aureus, coagulase negative na si S. epidermidis and si S. saprophyticus. Okay. So, laboratory diagnosis, how would we diagnose them? Okay. So, let's start with specimen collection and preparation. Okay. So, this is one of the ways we can collect for our sample. Okay. So, wet the cotton swab with NSS and use it to collect bacterial sample from the skin, nasal cavity, or facial swab. Or kung saan man yung kailangan nating, or kung saang part naman yung gusto nating mag-isolate ng bacteria. Okay? So, using the same cotton swab, inoculate the sample in a nutrient broth and incubate the tube at 35 degrees Celsius for 4 hours. Okay? So, 4 hours lang, gusto lang natin dumami ng konti pa yung bacteria para kapag nag-subculture tayo, more chances na may ma-isolate na tayo. Okay? Kaya, medyo maikli lang yung incubation natin. Yun lang yung purpose natin doon. Parang gusto lang natin medyo dumami lang ng konti yung bacteria na nakakuha natin sa swab. So, after that, so, incubate mo na. Okay? So, pwede na tayong mag-culture sa plates. Okay? So, using a disposable sterile wire loop, subculture the bacterial suspension in nutrient agar, BOP, and MSA. Okay? Yeah. And then, incubate the agar plates at 35 to 37 degrees Celsius for 18 to 24 hours. So, after the incubation period, observe natin yung bacterial growth. Take note natin yung kanilang colonial morphology, hemolytic pattern, and color development. Okay? So, for example, let's see the next slide. Yan. Okay. So, these are example results ni SRUs sa different plates. We have SRUs on MSA. So, ayan. Still, golden yellow yung kanyang color. On nutrient agar and on BAP naman. Ito. SRUs. Okay, next. So, hindi lang enough na i-culture natin siya. Siyempre, although we have an idea na golden yellow siya sa natumubo, hindi naman natin pwede sabihin na siya na yun agad. So, we have to test it pa to confirm. Okay, so one thing na kailangan natin gawin palagi is gram staining. Okay, so sa gram staining, so alam niyo na yung procedure niyan. So, yung expected result na lang, so since Staphylococcus, so that is gram positive, usually in clusters. Okay, ayan, yung itsura niya. Okay, pwedeng single, cocci, pero usually in clusters sila. Okay? Okay, so let's talk about MSA. MSA stands for mannitol salt agar. Okay, so this is a type of differential media na ginagamit natin for differentiating Staphylococcus species. Okay? So MSA have a high salt concentration of 7.5%, which inhibits most gram-negative and gram-positive bacteria except Staphylococcus species. Okay? Okay. So mannitol fermentation produces acid products, and the lowered pH changes the color of phenol red to yellow. Okay. So yun yung nagde-differentiate sa kanila. Kung sino yung mannitol fermenter and yung non-mannitol fermenter. Okay. So yung color indicator na kasama ng MSA is phenol red. Take note of that po. Okay? Yun. So, sino ba yung mannitol fermenter sa kanila? So, mannitol fermenter natin is si SRUs. Okay? So, you can see that sa naging kulay ng agar. Okay? So, normally, reddish in color yung ating MSA. Once na ay once ginamit natin siya to isolate SRU, so nagbabago yung color niya from red to yellow. Okay? So, that shows na mannitol fermenter si ating bacteria. Okay? So, which is a characteristic of SRUs. Si S. epidermidis, ayan, nakikita niyo yung colonies, pero hindi nagbago yung kulay. So, non-mannitol fermenter siya, pink colonies. Okay? So, catalase test. Catalase test is one of the tests we can use to further identify our Staphylococcus species. So, ang purpose nito, so this test, ayan, differentiates catalase positive, micrococcal, and staphylococcal species from the catalase negative streptococcal species. Okay? So, used most commonly to differentiate members ng catalase positive micrococcaceae from catalase negative streptococcaceae. So, some variations din, we can use this for identification ng mycobacterium. Okay? So, modified catalase test. Ginagamit natin for mycobacterium species. So, what is the principle of our catalase test? Okay. So, aerobic and facultative anaerobic organisms produce two toxins during normal metabolism. That is when oxygen is present. Okay, so ano yung toxins na yun? That is hydrogen peroxide and superoxide radical. Okay? Now, hydrogen and superoxide radical is toxic to the bacteria. Okay? So, how do bacteria protect themselves? They have what you call the catalase. Okay? So, catalase is an enzyme na present sa mga bacteria na to counteract the hydrogen peroxide and superoxide radical. So, ang ginagawa ni catalase, kino-convert niya si hydrogen peroxide to water and oxygen. Okay. Now, if walang catalase or walang catalase enzyme, yung bacteria, hindi niya kayang mabuhay in the presence of oxygen. Okay? So, yun yung mga strictly anaerobic organisms. Okay? So we have two methods. First, the slide method. Okay. So using a loop or a sterile wooden stick, just transfer a small amount of colony growth to the surface of a clean, dry glass slide. Okay. Then place a drop of 3% hydrogen peroxide onto the medium and then observe for the evolution of oxygen bubbles. Okay. So ito po yung positive natin, positive reaction and negative reaction. Okay. So positive natin, we have gas bubbles equals catalase positive po siya. Kapag hindi siya nag-bubbles, catalase negative siya. Another term for this is effervescence. Pag nagkaroon ng presence of effervescence, catalase positive siya. Okay? That is the slide method. So, pinakamabilis. Pero also, we have, so here is a sample video ng catalase test. Tala.
Today we're going to be doing the catalase test, and what it is if an organism is catalase positive, it can break down hydrogen peroxide into water and oxygen. So a catalase positive reaction is going to cause bubbles, whereas a catalase negative reaction is not going to have any bubbles at all. So what I did was I labeled the organisms out of the first grouping of organisms we use in class, which are positive, and those that are negative in regard to gram-positive cocci. So we have the Staphylococcus and the Micrococcus that should be positive, and the Enterococcus and the Streptococcus, which should be catalase negative. Now, if you look at the gram scheme of things, most everything is catalase positive except for Strep and Enterococcus. But in the first grouping, we have the gram-positive cocci, gram-negative cocci, and gram-negative cocobacilli. So that would be Staphylococcus, Micrococcus, Enterococcus, and Streptococcus are all gram-positive cocci. Then we have Neisseria, which is gram-negative cocci, and Moraxella catarrhalis, which is the only gram-negative cocci in Moraxella, and Haemophilus, which is gram-negative cocobacilli. So I put an organism on each that should be one that should be positive on here. So I put Staphylococcus aureus on here, and I put Streptococcus agalactiae on this one. So I'm going to add my hydrogen peroxide and look for bubbles. Okay, so as expected, the Streptococcus does not show any bubbles. On the video it looks really white, but there aren't any bubbles. Whereas on the Staphylococcus aureus, look at all those bubbles. Okay, so this is a positive test here for staph, and this is a negative test here for strep. Today we're going to be doing the catalase test, and
Okay, so another method na we can use is the slant method naman. Okay? So using a sterile technique, inoculate each experimental organism into its appropriately labeled tube by means of a strict inoculation. And then incubate them for 24 to 48 hours at 37 degrees Celsius. And then after that, allow 3 or 4 drops of the hydrogen peroxide to flow over the entire surface of each slant culture. So, ang titingnan natin dito is also the absence or the presence of the gas bubbles. So, ito yung positive result natin. Nagkaroon siya ng bubbling. And then, yung negative result natin is parang wala lang nangyari. Okay? So, yun yung ating slant method. So, what are the limitations of our Catalase test? Number 1, hydrogen peroxide is unstable and should undergo a control check daily prior to use. Okay? So, if parang sira na pala yung hydrogen peroxide mo na nagamit, so hindi na siya magre-react properly. So, false negative ang kalalabasan ng inyong test. Number 2, growth for catalase testing must be taken from an 18 to 24-hour culture only. Okay? So, organisms lose their catalase activity with age. Okay? So, resulting in a false negative reaction. Number 3, catalase activity is a function of aerobic process. Okay? So, organisms incubated anaerobically must be exposed to atmospheric oxygen for a minimum of 30 minutes before a catalase test is performed. Okay, so failure to complete this step may produce false negative results. Number 4, a positive catalase reaction with anaerobic organisms may be delayed for up to a minute after addition of the reagent. So if binasa mo agad siya or hindi ka naghintay na kahit saglit for anaerobic organisms, baka ma-read mo is false negative siya. And then a weak catalase or pseudo-catalase reaction may be produced by some strains of Aerococcus species and Enterococcus species. Okay. So this reaction is not a true positive test. Okay. Ayan. Pag weak catalase or pseudo-catalase reactions. So, bakit siya hindi true positive test? Kasi, ang tinatest natin for the catalase test is the presence of the catalase enzyme, di ba? Yung Aerococcus species kasi and the Enterococcus species, they have peroxidase. Okay, so peroxidase slowly catalyzes the breakdown of hydrogen peroxide. Okay, and last one naman is false positive may occur if the sample is contaminated with blood agar. Okay? So, it is best if kumuha tayo ng colony for testing sa nutrient agar na lang. Or kaya sa MSA, pwede din. Wag na lang sa back. Okay? Okay. So, what are the precautions naman na kailangan nating tandaan? Okay? So, dispose the hydrogen peroxide slides in the appropriate container filled with disinfectant. Okay? So, for slides used in bacteriology section, sa sharps din natin siya nilalagay. Okay? And then, yung sharps container nyo, merong disinfectant. Kunyari, may bleach solution sa loob. Para paglagay nyo doon ng slide, madi-disinfect na siya. Yan. And then, nichrome wire should be used when testing the organism. So, platinum wires may cause a false positive reaction. Then, when using a slant for other purposes in the same laboratory period, it is possible to save material by adding hydrogen peroxide to the slant after finishing with it. Okay, kumbaga, kung slant method na gagamitin niyo, gamitin niyo muna yung slant for other tests before kayo mag-catalase. Kasi once na nalagyan nyo na siya ng hydrogen peroxide, hindi nyo na siya magagamit, yung mga colonies. Okay? And then, number 4, extreme care must be taken if a colony is taken from a blood agar plate. Ayun. Okay? Kasi, erythrocytes or the RBCs, they contain catalase. Okay? So, a transfer of only a few blood cells, kahit konti lang, can give a false positive reaction. Okay? Kaya, as much as possible, iwasan na lang natin. Pwede tayo kumuha sa other plates. Nutrient agar na lang. Yan. Now, number 5, do not add organisms to reagent. Okay? Ano daw? Lang. Do not add organisms to reagent, particularly if iron-containing inoculating loops are used. Okay? So, it should be the other way around. Diba? Organism muna yung ilalagay natin sa slide, and then tsaka yung reagent. Hindi reagent muna, tsaka organism. Yun. Okay? Yan. Iron-containing loops also causes false positive result. Okay? Kapag na-expose sa hydrogen peroxide. Yan. Next, we have coagulase. Coagulase test. Sorry. Coagulase test. Okay. So, the purpose of our coagulase test is to differentiate Staphylococcus aureus, which is positive, from coagulase negative staphylococci. Okay? So, ano bang principle ng coagulase test? Okay? So, coagulase test, we test to detect the presence of the coagulase. So, coagulase is an enzyme na present kay Staphylococcus aureus that enables them to clot blood plasma. Okay? Similar to the mechanism ng normal clotting natin. Okay? So, ayun. So, if you've watched yung, sinit ko lang, Cells at Work, kung nanonood kayo ng anime, Cells at Work, baka familiar kayo dito, sa scene na to. Yeah. So, singit ko lang ito. If you haven't watched yung Cells at Work na anime, so, ito si Staph aureus, and this one, yan, sila yung white blood cells. Okay? And then, ito, yung shield niya, stands for the fibrin mesh. Okay? So, yun yung characteristic ni Staph aureus. Okay? So, in the presence of coagulase, it clots the blood. It clots our blood. Okay? Then, using that clotted blood as a shield. Okay? Now, protected na siya or hindi na siya basta-basta mapapatay ng ating white blood cells and other immune responses natin. Okay? So, yun. Singit ko lang. Gusto ko lang. Anyway. Yeah. So, back to this. So, the coagulase is described as a virulence factor. Okay? Pag sinabing virulence factor, it is a disease-causing factor. Okay? So, parang pathogenicity ng bacteria. Okay? So, one of the reasons kung bakit siya nagkakos ng infections. So, since kaya nga niyang mag-form ng shield or ng barrier, so, mahirap siyang patayin ng ating immune response. Okay? Kaya nakakakos siya ng infection. Yeah. Yeah. So, yung principle here, medyo confusing. So, mas madaling intindihan kung isi-simplify na lang natin, which is yung sinabi ko kanina. Okay, so, basahin nyo na lang this. Ito. Okay. So, most strains of S. aureus produce one or two types of coagulase. So, we have free coagulase and bound coagulase. So, free coagulase is an extracellular enzyme which reacts with prothrombin and its derivatives. Okay, so bound coagulase naman is localized on the surface of the cell wall and reacts with the alpha and beta chains of the plasma fibrinogens to form a coagulate. Okay? So, free coagulase is an enzyme that is secreted extracellularly, and bound coagulase is a cell wall associated protein. So, yung free coagulase, ang ginagamit natin sa kanya not method to detect the free coagulase is yung tube coagulase test. So, si bound coagulase naman is heat stable, again, localized on the surface of the cell wall, yan, and can be detected in the slide coagulase test. Okay. So, what is the method or procedure natin? Okay, so, for the coagulase test, place a drop of rabbit's plasma to a clean, grease-free glass slide. Okay. So, place a drop of saline. Na-confuse ako bigla sa pronunciation. Next to the plasma to serve as your control. Okay? So, get a bacterial colony of SRUs and emulsify in each drop of saline and rabbit's plasma. Mix well for 5 seconds and observe or take note for clumping within 10 seconds. Okay? Record and illustrate the result. Pag nagkaroon ng clot formation or clumping, that is positive for coagulase, slide coagulase test. And then kapag wala naman, edi negative for slide coagulase test. So let's watch this one.
Coagulase test. The coagulase test is used to differentiate species within the genus Staphylococcus. The test indicates the ability of an organism to clot plasma by the action of the enzyme coagulase. Using a transfer pipette, add a drop of coagulase plasma reagent to a clean slide. Then add a drop of water to use as a control. Using a sterile toothpick, remove an isolated colony from a blood agar plate. Mix it into the coagulase reagent. Always include a control using the remainder of the selected colony dispersed thoroughly in a drop of water. A positive result is identified by the clumping of organisms after 1 minute. The control should remain thoroughly dispersed with no clumps. The control must show this result before the test can be read.
So for the tube coagulase test naman, so put 0.5 ml of rabbit's plasma in a 3 ml tube, and then get a loop full of your bacteria and inoculate them in the 0.5 ml of rabbit's plasma. Incubate at 35 to 37 degrees Celsius for 4 hours, and then check the tube every 30 minutes to take note and observe for the presence of gel-like clot. Okay? So if no inoculation is formed, incubate the tube further up to 18 hours. Okay? So, yun. So, una, 4 hours natin siya i-incubate, pero i-check natin siya every 30 minutes. Okay? So, bakit kailangan natin i-check every 30 minutes? For the presence of the gel-like clot. Okay? That is because of the tendency na mag-autolyze yung gel-like clot because of the presence of staphylokinase. So, staphylokinase is also an enzyme present in some strains of Staphylococcus. Okay? Yun. So, we need to observe them every 30 minutes kasi may tendency na mag-autolyze or matunaw lang din ulit yung gel-like clot because of the presence of staphylokinase. And then, if wala talaga, for every 30 minutes under the 4 hours, further i-incubate pa natin siya up to 18 hours. And then, i-check natin siya sa dulo kung wala talagang na-form. Okay? So, nagkaroon ng gel-like clot, it was positive. Pag walang gel-like clot, that is negative tube coagulase. Yeah.
one of the most useful tests for differentiating Staphylococcus aureus from other species of Staphylococcus is the coagulase test. The coagulase test looks for the enzyme coagulase, which causes citrated rabbit plasma to gel or coagulate. The enzyme coagulase causes soluble fibrinogen to be converted into insoluble fibrin. And Staphylococcus aureus are often referred to as coagulase positive staphylococci because they produce the enzyme coagulase. Most other species are referred to as coagulase negative staphylococci because they don't produce that enzyme. So this is a tube of citrated rabbit plasma that was inoculated and incubated. This is a coagulase positive bacterium. We'll see that the rabbit plasma at the bottom has gelled. So that would be coagulase positive. And this is a coagulase negative bacterium. We see that the rabbit plasma remains a liquid. It hasn't gelled. So it's pretty easy to recognize a coagulase positive bacterium because of the coagulation of the rabbit plasma. Now in a lab, we're going to be doing this using these small centrifuge test tubes, but these are a little harder to see the results on when I try to video it, so I've showed you the results in a tube rather than in the small tubes that we'll be using in a lab.
So, what are the limitations naman ng ating coagulase test? Okay? So, the slide test should not be performed with organisms taken from high salt media, such as mannitol salt agar, as the salt content can create false positive result. Okay? So, kung yung kanina, catalase, kailangan natin iwasan yung blood sa blood agar, this one naman for the slide coagulase test, huwag tayong kukuha sa mannitol salt agar. Okay? So, pwede tayong kumuha sa nutrient, agar, sa BAP. So, overmixing naman may cause the clot to break down. Okay? So, dapat, hindi ma-over sa mix. Yun. The tube test is more reliable than the slide test. And then, we generally don't use the coagulase test when identifying unknowns. Okay? And then, samples must be observed for clotting within 24 hours. Okay, so this is because some strains that produce coagulase also produce an enzyme called fibrinolysin, yung sinabi ko kanina, which can dissolve the clot. So therefore, the absence of a clot after 24 hours is no guarantee that a clot never formed. Okay? So the formation of a clot by 12 hours and the subsequent disappearance of the clot by 24 hours could produce a so-called false negative if the test were only observed at that 24-hour time. Ayun. Kaya natin siya ino-observe for every 30 minutes for the first 4 hours and then further i-incubate pa natin siya, pwede rin natin siyang i-check from time to time. Hindi lang isang beses, hindi lang sa 24-hour or sa 18-hour mark. Okay? So, summary. Okay. So, atong table na to, I suggest you memorize them. Okay, para hindi tayo nalilito. Okay, so for kada catalase test, all of them are positive. Staphylococcus, usually, in general, are positive sa catalase test. However, we can differentiate them na through coagulase test. So, coagulase, ang only positive coagulase natin is si S. aureus lang. Okay. Growth on MSA, si S. aureus lang ang mannitol fermenter. Yung dalawa, though change in color. So, they are not mannitol fermenters. And then sa novoviocine susceptibility, novoviocine is a antibiotic. Si Saprophyticus lang ang resistant. Okay? So, yon after each laboratory, as usual, cleaning ng glasswares and everything, disposing of your wastes. Ayun. So, alam nyo na yan. Okay? Yeah. And then, thank you po. Okay. So, for our activity nito, I will be posting na lang po yung step-by-step procedure and then yung instructions for the activity. Okay? So, thank you po and yun, God bless.